Welcome to the last class of the first colloquium in embryology. This class will be the most advanced and most complicated class because of all of these very, very confusing slides that we have here. It's also going to be the class that is most important in preparation of colloquium 2 as well, even though it's a colloquium 1 class. These are structures that will reoccur during your studies for the colloquium 2 of embryology. We're going to start with the developing vertebral column, so we're going to skip this for now. And one of the most important things that I can recommend before for studying this class is that you'll have to use one of the most important words right before each and every structure, specifically for this class. And that word is development or developing. So because we're dealing with so many structures that aren't fully developed or are in the process of being developed, we're going to have to use this word before the actual structure name in order to gain all of the points we want for the practical part of the colloquium. So if you don't use development of the vertebral column and you just write vertebral column, you might get not gain all of the points you want or full points for that. When do we use development? We use it specifically for structures that aren't specific for that stage of the embryological development. For example, do we have a vertebral column when we're grown up? Yes, we do. We actually very much do have a vertebral column as adults, right? So therefore, during embryology, this will be a developing vertebral column. Do we have a notochord when we've grown up? No, we don't. So therefore, we don't have to write developing notochord before each and every structure name. Same thing goes with no, I think that's it. Okay, perfect. So again, remember, please use developing when we're dealing with structures that we actually do have as adults. You don't have to use them every time, but you have to use it very often. So yeah, let's get started with the actual vertebral column. Now, the best way I can describe the vertebral column is if you imagine bricks followed up by spackle in between bricks. So if you've ever seen bricks before in your life, I hope you have. You have these red bricks, right? And then you have the gray spackle in between, followed up with another brick, and then additional spackle, and then another brick, right? That's kind of how what the general idea of the vertebral column is. So if we look at the brick or the developing vertebra, we can kind of see it here. So this entire thing is that one brick or a developing vertebra, and then the spackle is the intervertebral disc, or of course developing intervertebral disc. So you can see it all here. So it is what separates each and every developing vertebra from one another. So all of these are separated by this spackle called the developing intervertebral disc. Why developing? Because we have intervertebral discs of our own and we have vertebra of our own, so we have to write developing. Professor Rona did say that you can also write resting hyaline cartilage here. You don't have to use developing before because hyaline cartilage is very tied to embryological development. So check that with your professor. I would go with the developing vertebra because it's just an easier structure to remember since we're looking at the vertebral column. Followed up by that, we have a more unique area or structure where we don't have to use developing, thankfully, and that is the primary ossification center. Now, you might remember this from histology colloquium 2, I think. So in the center of these big bricks of hyaline cartilage, we have these darker areas filled with hypertrophic chondrocytes. Now, and they are called the primary ossification center. They are developing this matrix that appears as purple and help with the full development of this cartilage. So in the microscope, they are very, very visible. How do you say it? They are very easy to spot because of the fact that they are such a huge structure within this entire vertebral column, I would say, or vertebral, or just vertebra in general. Okay, those were the primary ossification centers. Now, if we look at a microscope in regards to the primary ossification centers, we can actually see the chondrocytes as well. Let me look at it with only the images. Here we go. Now, this is what it might look like under the microscope, a vertebral column, that is. And although it appears a lot lighter and less distinct, you can still see the corners of these developing vertebrae separated from the developing intervertebral discs. I hope they are visible here. And then you can also see the processes of the vertebral columns. Here you can see it as well. This would be the intervertebral disc. Hopefully it appears more, or appears well in this YouTube video. And then here you can see the actual vertebra. And then in the middle, such a characteristic shape and color, we have the primary ossification centers. Now, this is just a zoomed in version here. And here you can actually see the hyaline cartilage, and then the center called the primary ossification center with a matrix. You don't have to write in matrix, but you might be asked about one of these large cells that makes up this primary ossification center. And these cells are called chondrocytes. If they are large and bloated like this, you might have to write hypertrophic chondrocytes, but just double check that with your professor. Again, if we're inside the the vertebral column and we're looking at the center of a vertebral disc, something like this, and it looks like it has like a very dark purple opening, almost like an eye shape, sometimes not an eye shape, just an opening. We zoom in, they ask you about the entire structure, its primary ossification center, and then if they ask you about just a cell, that could be a chondrocyte, alternatively a hypertrophic chondrocyte. All depends on what your professor prefers. Following that, we do have another area that kind of looks like a primary ossification center, just without the color, I would say. So we have the remnant of notochord. Now you might remember notochord, the chorda dorsalis, from the germ layer. Should I show it? Let's show it. It's easier to remember that way. And I think it's here. Yeah. You might remember the notochord from here. Now the notochord has finally degenerated and it is appearing like this instead. So I would best describe the visual characteristic of this remnant, you have to write remnant of notochord, as if you ever imagine like tears in a nylon stocking. So in this case, the nylon stocking would be in the entire vertebral column, and then if we have a tear in it that appears like this, could be in the intervertebral disc, could be in the hyaline cartilage itself. It is the remnant of notochord, like sort of like a rip inside of this whole area, right? We can see it in a demo lab slide. Here we go. So again, a little bit less HD quality, but hopefully it makes sense. This could be a remnant of notochord because it looks like it has ripped open here. But more importantly, if we look at this primary ossification center and go further down here, we can actually see one that is almost perfectly ripped open here. This might also be it. I don't think so because of the color of the of the actual spot. It might still be it, but I would say this looks more like a remnant of notochord. Could I see this could be a remnant of notochord? This could also be one. More importantly, if we look at the slides, like I said, a rip in the texture of the entire vertebral column, very characteristic of the remnant of notochord. You can see it here too. This could also be a remnant of notochord. This as well, maybe not because of the like very abundant purple inside. Again, if it's a rip that has all of these dark purple extracellular matrix in it with the chondrocytes, then it's definitely the primary ossification center. While if it just appears as a rip, it is the remnant of notochord. This could also be remnant of notochord. Okay, and hopefully you remember from histology colloquium 2 that a hyaline cartilage is surrounded by the perichondrium. So the layer surrounding each and every one of these vertebra is known as the perichondrium. And it exists everywhere. It's not that important of a structure, but it is important, but it could be good to remember that from previous colloquiums. Okay. Finally, we have two adjacent structures that always surrounds the vertebral column, even if it's separated like this. So the first structure is this tissue that is always close by to the vertebral column. You can see it here. It appears as fabric that is sort of swaying around the vertebral column. And these are just mesenchymes made up of the mesenchyme cells. So this is mesenchyme as well. This could be, that is more likely a part of a ganglion. That is also not as well. This could be mesenchyme as well. More importantly, there is a structure that almost always appears close to the vertebral column and is very important for your colloquium, and that is the developing spinal ganglion. Again, we use the word developing before because we have spinal ganglions as adults. Now, the developing spinal ganglion, I best describe it as looking like a jellyfish, or a kidney of sorts, or like a balloon, right? It has this dark body. I think that you should remember how a spinal ganglion looks, or what a ganglion looks in general, nerve ganglion. And it has these lower processes that extends outward. Now, this is how it looks like here. Again, the dark circular purple body with the tentacle-like structure that follows it, like so. We can also see it here. We don't see the lower half, but definitely if you see something that appears this round in shape and purple next to the vertebral column, 9 out of 10 times it's the developing spinal ganglion. You can also see it kind of here, just a little bit fragmented. This could also be it. And then in the context of a sagittal section of an embryo, all of these rounded, darker splotches, also this, are developing spinal ganglions because they are next to the developing vertebral column. I would say even these appears like it. But again, very easy to identify. They just look like a hot air balloon or a tree maybe with a stem and a crown, right? I think that's it for the separate area of the developing vertebral column. You might not need to know about the humerus development for this colloquium, but do double check that with your professor. Okay. Yes, so now we can finally get into the development of the vertebral column in the context of embryo in sagittal section. Now, we received the structures of the developing vertebral column, but in the context of an embryo cut in a sagittal section. Now, how do we see that it's the vertebral column? First of all, it appears as a snake or a worm that has all of these different parts to it, right? Very easy and very characteristic of a vertebral column. Next is that it doesn't appear as fully circular or oval like a developing rib. This is another structure that might appear. The developing rib and the developing sternum are always round or oval, and they don't extend that much. Oh, this is the developing sternum, excuse me. They're always round and they don't extend, but they follow the thorax like so. While the developing vertebral column might appear as round at some parts, like here, but this could be just the processes and this could be the actual body of the vertebral column. So please do keep that in mind, that a lot of students make the mistake of mistaking the ribs from the vertebral column. Okay. Also, the developing ribs stop at a certain point right before the diaphragm, while the developing vertebral column continues further down into the body of the embryo. This is also not the head, even though it appears that this is the head of the embryo, it's not. This is actually the bottom, and the head would be somewhere here. Okay, so in terms of the vertebral column, again, do not mistake it for the developing ribs, and also it's always followed by these developing spinal ganglion. Now they might ask you about just one developing vertebra, or like I said, the spackle in between, which is the developing intervertebral discs. Here you can also see a remnant of notochord because it acts as a tear in the middle of the developing vertebra. And we can't really see the primary ossification center in this case because we don't have a center here that is sort of bubbling and frothing and very, very dark pink or purple. With the chondrocytes, mainly we just see the remnants of notochord. Here is another section of the vertebral column. I'm zooming in because this is how it might look like in the colloquium, or what it might look like. And again, we have the remnant of notochord, the vertebral column. Maybe we should look at it from a non-annotated perspective here. So again, we have the vertebral column here. Why is it a section like this? Well, this might be the processes, this might be the body of the vertebral column. That's just why it appears as if it's divided in two. We have a remnant of notochord here because it's a little split area. More often than not, in these full embryo sagittal section slides, you'll probably more often see the notochord or the remnant of notochord, rather than the actual primary ossification center. Followed up by the surrounding these like dark purple orbs, which we know as the spinal ganglion or developing spinal ganglion. So that's, and again, it's easy to differentiate the vertebral column from the developing ribs because the developing ribs are circular in shape and are not followed up by something else. This is the sternum. This is the sternum. While the developing vertebra, while they might appear as circular at some points, usually come like this, right? So should be easy, fairly easy to identify. Also, the sternum ends right before the diaphragm begins. It's primarily composed of the thoracical area or section of the embryo, while the developing vertebra continues further down. In this case, it ends also kind of before the diaphragm. Okay, let's continue here. Here we can also see the developing vertebra. And more importantly, we can actually see the primary ossification center. So you can see here we have a very identifiable line of the developing vertebra. We can also see it in sections here. While here we have the developing ribs because they are more circular in shape, right? And uniquely, here we can actually see the primary ossification centers, which appear as these static, like have you ever seen the a static television with the black and white dots. That's kind of how it appears here. Zoomed in, it will look more like a normal primary ossification center. You can see it here as well. But we can also see the remnants of notochord, which are also very, very big like this. I believe that would be the remnants of notochord. They are always in between these intervertebral discs, apparently here, and one of the vertebral columns as well here. Do we see any spinal ganglion? Can I spot any? This might be spinal ganglion, but it also might be part of this structure, which is the nodal tube. So I am too afraid to tell you what. I can't really find one that is very identifiable in this case. But again, very, very clear spinal or vertebral column. Okay, and here we can see it as well. Again, like I said, it might appear as circular, but if it's followed up by these areas, you know that it's the vertebral column. Well, the rib cage only has these splotches of circular hyaline cartilage, but it's not followed up by another section like this. So this might be the body, or this might be the processes, and this could be the body of the spinal or the vertebral column. Here we can actually see the spinal ganglion. So this is, for example, one. This could be another. This could be another as well. In this case, we don't see the intervertebral discs that well, but we do see the separate vertebral columns. So they might ask you about that instead. I think that's it for the sagittal section vertebral column. We'll come back to it when we review the entire embryo and sagittal section as well. But let's continue on with other structures now. Now, a closely related structure to the vertebral column, although it might not appear like it, are the developing limbs of the embryo. So we can see one here, we can see one here. Now, in the microscope, they won't be this zoomed out, obviously, and you might not actually be able to identify what kind of limb it is. In this case, we know that it's the upper limb because we can see these clear fingers here, right? Well, here we know that it's the lower limb because we can see the knee and the kneecap areas as well here. But things that it has in common with the vertebral column are the developing hyaline cartilage, or not developing hyaline cartilage, but the developing bone, aka the hyaline cartilage. Now, the same thing with the vertebral column, we have a let me double check, we have a perichondrium, we have a perichondrium, there we go. Perichondrium that follows all around this hyaline cartilage or developing bone. You don't have to be specific with what part of the bone it is. Again, because of the fact that it's going to be super zoomed in the colloquium, I doubt the professors are going to be that harsh to you. They weren't for us, at least. Again, though, ask your professors. Please ask your professors before. Myotubules is usually not important for this colloquium. It was brought up during our practical classes, but it wasn't covered by the list of structures, and therefore the myotubules weren't important at all for this colloquium. However, still might be important for the next course or for you guys. So check that first before doing anything. Like I said, following hyaline cartilage, we have all of this tissue like we had with the vertebral column here. We had this loose tissue that kind of looks like loose irregular connective tissue, right? But in embryogenesis, this tissue is simply known as the mesenchyme. So if you see any sort of section of a tissue that sort of looks loose, sort of looks like it looks like CT, know that you can just write mesenchyme, and usually you'll be right. However, we do have another type of tissue that is very important for these types of structures, and usually the type of tissue that is focused on during the practical part of the colloquium. And those are the immature skeletal striated muscle fibers. Now they appear, like the name suggests, as muscle fibers. How can we see the difference between the muscle fibers and the mesenchyme? Well, the muscle fibers appear, like the name suggests, as fibers. It goes sort of like in a longitudinal section. Sometimes it can be cross-cut, but you can see here that it looks like small steps or lines that goes up. Now that is very characteristic of muscle tissue. Have you ever just gotten like a piece of steak or meat and you've cut it in a specific section, and you can see all of those fibers in a longitudinal cut? That's sort of what you see here as well. Again though, for this colloquium, you'll have to write, instead of just writing muscle fiber, you have to write the immature before, because we're dealing with embryogenesis, so it's not developing in this case, it's immature. Skeletal striated, which is the type of muscle fiber, and then of course muscle fiber next to it. So this entire name is important, and it's the case for all muscle fiber that is surrounding a type of hyaline cartilage structure. You just write this, right? The smooth muscle cells you'll only see in developing or not developing in human uterus, which is already fully developed. We're looking at an adult human uterus and adult prostate as well. You'll see smooth muscle cells here. In all other cases, if it's close to hyaline cartilage, it's most often the immature skeletal muscle fibers. So that was that limb. We have it here as well. Here you can actually see those lines of the immature skeletal striated muscle fibers more closely here. You can see it here as well. See how they form these longitudinal sections? Those are very characteristic of muscle fibers. Here instead we have the developing bone or hyaline cartilage, this entire thing, the perichondrium which surrounds it, right? And then we have the chondrocytes, and then calcification of chondrocytes. Might not be important for this colloquium, but do keep that in mind, that some structures from colloquium 2 in histology might appear here as well, especially in terms of hyaline cartilage and blood vessels as well. But like I said, we have this loose tissue here that kind of looks like loose irregular connective tissue, and for us, because we're in embryogenesis, it's the mesenchyme, this and this, right? But any other tissue that appears as muscle that is close to hyaline cartilage will be the immature skeletal striated muscle fibers. So in this case, it's very beautifully apparent here that all of this would be the immature skeletal striated muscle fibers. If they ask you about a section of it, then instead of writing fiber, you'll have to write fiber bundle. So it's several, it's plural. In this case, I only wrote fiber. I'll change that hopefully before sharing it. But here you'll actually have a bundle of fibers, right? One fiber would just be one line. But a bundle would be this entire section here. Well, if they ask you about just the fiber, they would zoom in and show you just one fiber, like so, and ask you about the name, and in that case it would be just singular fiber and not the bundle. So plural, if there's several, we add a bundle here. If it's just one, we add the fiber. We're gonna zoom in more with the skeletal striated muscle fibers, or immature skeletal striated muscle fibers, to show you guys examples of it. So again, now more in detail of immature skeletal muscle fibers and its structures. So like I said, these fibers usually follow close to the hyaline cartilage, like close to a limb, close to a vertebral column as well, a hand, a leg, anything, we'll probably see a muscle fiber close by. We will also see mesenchyme, which appears as this loose, irregular connective tissue-like structure, like so. So zoomed in, the immature skeletal muscle fibers appears like this. Remember when I said that if you see these long, longitudinal cut sections, this would be very characteristic of the immature skeletal striated muscle fiber bundle. Well, if you see this loose, irregular connective tissue appearing area, that would be more like mesenchyme. So here it is more zoomed in, but well, we'll get to that in a second. And we have two types of cells in relation to the immature skeletal striated muscle fibers. The first one is the myoblast. Let's see here, myoblast. And they have a more pink and thicker cytoplasm that is very visible and a very large nucleus. So if we see it here, we can actually see it kind of here. So here we have a thick nucleus and then a pink cytoplasm, almost like a crescent moon shape surrounding it, right? So that would be a myoblast. Here's another myoblast, so a thick nucleus and a pink cytoplasm surrounding it. This could also be a myoblast. More importantly, I think we found one here. Here we go. See here, this is a more identifiable myoblast. So here you have the thick nucleus and then the cytoplasm surrounding it, like so. Here as well, and then cytoplasm surrounding it, pink cytoplasm specifically. Here it is as well. Here it appears more rounder rather than eye-shaped. So here you can see the nucleus and the cytoplasm. Nucleus, cytoplasm, nucleus, cytoplasm. While the mesenchymocyte is the other type of cell, and it appears in two ways. The first way is that you can only see, let me actually draw over this. The first way is that you can only see the nucleus like this. So before we had the nucleus and the cytoplasm, right? In this case, we can only see the nucleus. The other way is that we see the nucleus and we see a very, very, very tiny, almost like a dot in the center, nucleolus. Now, a lot of people make the mistake of assuming that this is the myoblast because we can see what appears as cytoplasm with the nucleus. But in fact, this is the nucleus and this is just the nucleolus. So if the nucleus-like structure appears as very, very tiny within a very, very dark area, it's more often than not the mesenchymocyte. Now, what do I mean by that? Here we can see a great example. So here we have the myoblast that we talked about before with the cytoplasm and the nucleus in the center. The mesenchymocyte, on the other hand, is here. It appears like this with this tiny nucleolus in the middle. So see how tiny the nucleolus is here, the center, compared with the nucleus here? That's because this entire thing is actually the nucleus. It doesn't have a clear cytoplasm, and instead we can actually see the nucleolus. I think this could be the mesenchymocyte as well, with the tiny, tiny nucleolus in the center. But sometimes it just appears as the nucleus. We don't actually see the nucleolus in the center, like we do in this. Here we go. So here, like I said, the myoblast, which has a nucleus and then a surrounding cytoplasm, while the mesenchymocyte kind of looks like fibroblasts. So they appear like this, like an entire nucleus with no, in this case, it doesn't have the visible nucleolus in the center. This entire thing is the nucleus. You can kind of compare it with the human urethral epithelial cell and the lymphocyte from sperm cells, only here it's a little bit more difficult to see the difference. But if the professor does bring this up in your practical colloquium, they will make it very obvious which cell they're asking about. So in this case, this very much looks like a fibroblast. This does not, right? So this would be the mesenchymocyte, while this would be the myoblast. And all of these can appear close to the hyaline cartilage within the immature skeletal striated muscle fiber area. Yes, so here we have the immature skeletal muscle fibers, one here and one here. Like I said, if they asked you about this section, you will have to write bundle after the fiber. But if they only ask you about this section, then it would be enough to just write the fiber without the S. Okay, sometimes you can also see blood vessels. That might be like a little sneaky structure that might be included in your colloquiums. I think those are it for the muscle fiber. Let's look at them from a demo lab perspective. Let's see here. There we go. So here we can see the vertebral column, right? With the primary ossification center. Going down, these are all of in the same slide because they are, or maybe not, but let's just assume they are. But they might be in the same slide, right? And here we have a limb development. See how hard it is to differentiate if it's the upper or the lower limb? I would say upper limb, but I might be completely wrong, right? So here we have, actually it's probably the lower limb. Nonetheless, here we have the hyaline cartilage or the developing limb with the perichondrium surrounding it, and then in the center we have all of these chondrocytes. Here we have calcified chondrocytes. And then zooming in on this section, specifically this section, you can see all of these lines almost appearing like hair, right? And what did I say about these lines? Then we know that we are in muscle territory, right? Zooming in on that, if we will see something like this. Now here it doesn't look that muscle-like, but again, they will first show it to you zoomed out, and you will know that it's either muscle fiber or it's mesenchyme. In this case, if it appears as lines like this and it's next to bone development, then we know we're dealing with muscle. In this case, if they ask you about this entire section, the name for it would be immature striated skeletal muscle fiber bundle. Well, if they just ask you for one, you will remove the bundle word in it, and you will just write immature striated skeletal muscle fiber. There we go. And this is more likely to be blood vessels, maybe even capillaries. Here is more examples of the same thing. Now, they might ask you about the difference between a skeletal striated muscle fiber and a myotubule. Again, that's what they went through in the practical class, but it wasn't something that was brought up in the colloquium. In fact, it wasn't even on the list of structures. So it might not appear, but let me go through it just in case. A muscle fiber, should I do it? Yes, let's do it. A muscle fiber has the nucleus in the center adjacent to one another, so it looks like almost like a vertebral column stacked up like this. Here we can see it as well, but sort of segmented, right? While a myotubule has the exact same structure of a muscle fiber, only that the nuclei are separated and elongated like this. Maybe I can find a better example of this. I think this would be a good example. This would might be more of a myotubule with all of these nuclei separated like so and elongated, while this would be more of an immature skeletal muscle fiber with the nuclei being more in a vertebral column line like so. But again, might not appear in the colloquium. I just wanted to mention it just in case they bring it back. Okay, I think we figured out the muscle fibers. Now let's get into more of the organs of the embryo in sagittal cup. Before we get into the specific organs of the embryo in sagittal section, I really want to point out how important it is to identify the different sections of the embryo or total embryo in sagittal section. And why is this important? Because depending on the area that you're in, you're going to have different types of organs, and therefore you're going to be able to know what type of organ it is, even though they barely differentiate from one another. For example, the Metanephros might appear very much alike the lung in pseudoglandular stage, but if you know that you're in the thoracical area right here, and if you can identify that, you will know that, okay, well, this has a lesser chance of being the Metanephros because the Metanephros can only be in the abdominal area. Okay, hopefully that made sense. So first of all, how do we identify the abdominal area from the thoracic area? I want to point out that the slides are going to be very zoomed in, almost like this, right? So you're going to have to know the difference between the rib cage and the vertebral column in this case. Like I said before, the rib cage being a lot more rounder, the vertebral column being more snake-like and having all of these vertebrae composed of the intervertebral discs in between them as well. So for the thoracic area, we are following along the rib cage, and it ends when the rib cage ends. What do I mean by that? Well, if we look at this slide here, the blue area being the thoracic area, sorry. Yes, we can see the rib cage right here. We can see that they are very circular in shape, while here, even though the vertebral column also have these circular areas, it's followed up by other processes of the vertebral column. So we know that this is not the vertebra, even though, again, it has a circular, as long as it's connected or has this separate additional area of developing bone, we know that we're dealing with vertebral column and not the developing rib. The developing rib has only these circular developing bone areas or hyaline cartilage areas, with no following bone area, right? Okay, so in the thoracic area, we're gonna follow the developing rib. And as soon as the developing rib ends, we're gonna have this very distinguishing line that separates the abdominal area from the thoracic area from the abdominal area, and that is the diaphragm. And the diaphragm is a set of muscle tissue that separates the two all across like this. Sometimes it's we can only see fragments of the diaphragm. For example, here we're following the rib cage like so. So this is the thoracic area, right? And then right underneath the rib cage, we should have the diaphragm. However, here, even though we can see it start, it sort of ends here, and theoretically it's supposed to continue all around the other side like so, separating the thoracic area from the abdominal area. We can see it here as well. Here more in line with how it will actually look like with all of this skeletal muscle tissue, the diaphragm right underneath the rib cage. Now that's how we know that we're in the thoracic area. The thoracic area also contains these following important organs that we're going to get into later. The developing lung in pseudoglandular stage, do you have to write it all out like this? Yes, you do. Specifically the pseudoglandular stage name. This will also be brought up in colloquium 2. In colloquium 2, you'll have additional structures following within the lung, but in this case, in this colloquium, you only have to identify the entire organ, the lung itself, no structures within it, right? So here, for example, the developing lung appears in the thoracical region. It looks very much like the metanephros. We'll get into the difference between the lung and the metanephros in a second, later on in this video, but just to make it easier here, where is it? There we go. So the lung is one of the larger, obviously, organs that we'll have in the thoracic region. And then we also have the developing heart. Now, just because the thoracic area contains these organs doesn't mean that we're going to see them all the time. Sometimes we don't even see any of these parts, depending on the cut of the slide, of course. But in this case, we can actually see the developing heart right here. We'll get to the heart in a second. There are structures within the developing heart that we do need to know. Okay, other organs within the thoracic area is the upper dorsal aorta. We can't really see it here, but we do see it here. So this would be the thoracic area. Why? Because we're following the rib and it's separated by the diaphragm here, only it's segmented diaphragm here. We can see the lung, or the developing lung in pseudoglandular stage, and we see the developing dorsal aorta like so. How do we know that this is dorsal aorta? Well, it kind of appears as a super large blood vessel or a large opening like so, with all of these endothelial cells lining it with a large lumen in the center. That's how we know we're looking at the dorsal aorta. The next organ is the structure is the upper vertebral column. So even though we can only see the developing rib in the thoracic area, we can see the vertebral column both in the thoracic area as well as the abdominal area, right? Because it follows along like so. So we will be able to see the upper vertebral column in the thoracic area, just as we can see the lower vertebral column in the abdominal area. For the abdominal area, we are not going to be able to see the developing ribs. So we're going to go past the ribs. We might see like the outer belly area with the developing skin. I think that might be more of a colloquium 2 structure. But here the ribs are cut off and we're starting with underneath the diaphragm. We can see a very, very large organ here, known as the liver, of course. But this, again, is more of a colloquium 2 structure. We might see it, though, in colloquium 1, but we will not be asked about it. But it could be a good signifier of where we are within the sagittal cut of the embryo. We do see some structures. We see the Metanephros, the developing adrenal gland, Mesonephros, developing gonad, and developing hindgut. We will get to these structures in a second because these we actually need to follow through. But this is how they will all look like in the context of this slide. We will also see the lower dorsal aorta. So like we saw the upper dorsal aorta in the thoracic area, we will see the lower dorsal aorta in the abdominal area. Do you have to write lower and do you have to write upper? No, this is just for me to be able to explain it better. Here we go. So here we saw the developing dorsal aorta in the thoracic area, and it continues on the abdominal area, only we can't see it here. Here we can actually see it more clearly. So the dorsal aorta will start here and go further down, and here we can actually see it start at the abdominal area instead of the thoracic area. Okay, and also we do know that it is a dorsal aorta. We can look at the contents of it with the red blood cells. We can also see if it's lined with endothelial cells. And then finally we have the lower vertebral column. So this would be upper vertebral column and this would be the lower vertebral column. But again, very big signifiers, we can't see the developing ribs. Okay, those are the differences between the abdominal area and thoracic area. Now we can finally get into the organs. For the thoracic area, we will focus on the developing heart, and for the abdominal area, we focus on these four and slightly these organs or structures. Okay. Now we can finally start with the first official organ of the embryo and sagittal section, which is the human heart. Now the human heart can, like the vertebral column, come separate like this, or it can come inserted already into a cross-section of the total embryo. The human embryo heart can appear in two different ages. It's either 6 to 7 weeks or it's 10 to 11 weeks. There's also a 12-week-old heart further down, but these two are the main age that you have to be concerned with, because in the colloquium, if they ask you about the entire slide, you will also have to write down age, and in terms of the amount of weeks. I'll tell you the difference between these two in a second. Let's first divide the heart into two main sections. The heart has a top section known as the truncoconal region. Now this section contains the aorta, it contains the vena cava, and it contains the pulmonalis. So you can see it, and here it is sideways, but you can see all of this would be the truncoconal region, this would be the truncoconal region as well. And then it contains the body of the heart. Now the body of the heart has 4 chambers. I hope you remember this from physiology. It has the atrium, 2 of them, and then it has further down the big ventricles. Here in this particular slide, the ventricles are further up. So this would be the ventricles and these would be the atrium. Here we can't really see the atria because it's cut in a certain way where we don't include the atria. But now we can finally start with the difference between the ages of the heart. Hopefully this is the right, yep, okay, perfect. Here we see a 6 to 7 week old heart, and here we see a 10 to 11 week old heart. Remember that you have to write the magical words developing right before a structure that also appears in an adult human. So obviously we have a heart, and therefore you have to write developing heart and then 6 to 7 weeks in order to get the full points, depending on the slide that you're looking at. Now, for the 6 to 7 week old heart, like I said, we have 2 atria and 2 larger ventricles. Now these ventricles, not only do you have to write the developing ventricles and atria, you also have to write in a specific adjective to describe it if we're looking at a 6 to 7 week old heart, and that adjective is primitive. So in this case, these 2 would be primitive ventricles, and then this would be a primitive atrium. So primitive right before the chamber of the heart if it's the atrium or the ventricle, and of course you have to write developing before. In a 10 to 11 week old heart, instead of primitive we use the word primary. So in this case, a primary ventricle or a primary atrium. Now that's a theoretical difference in terms of what you write in the practical part of the colloquium. For visual differences, we actually have 2 signifiers that will help you identify if you're looking at a 6 to 7 week old heart or a 10 to 11 week old heart. The first difference is something that we can visually identify in a much older heart, which is the pericardium. The pericardium is almost like a loose area of tissue that surrounds the entire heart like this. You can also see it here as well. Almost like a shawl that surrounds the heart. In a 6 to 7 week old heart, we have no pericardium, so we have no such loose tissue that surrounds the heart like so. So that is one of the most primary differences between 10 to 11 week old heart and 6 to 7 week old heart. The other visual difference is what they look like in a microscope or under a microscope. For a 10 to 11 week old heart, the heart is so developed and too large for the microscope to actually capture the entire slide. Instead, we get sections of the heart like so, maybe even half of the heart like so. While in a 6 to 7 week old heart, we can actually see the heart in its entirety under the microscope because it's that tiny. Those 2 are the main differences between the ages. Now we can actually get into some structures that both share. Okay, here we go. Now, like we said, we have a truncoconal region of the heart. There we go. And we have the main body of the heart, as well as the top of the heart, or the tip of the heart, known as the apex cordis. The heart is also made up of a certain kind of tissue. This tissue is called the myocardium. And the myocardium is also made up of specific cells known as the developing cardiomyocytes. So if they ask you about a section of the heart that looks just like tissues, something like this, you will write in myocardium, or developing myocardium would be more accurate, I would say. And then if they ask you about a specific cell, it would be the developing cardiomyocytes. You can see it here. I didn't write in the myocardium, but if they ask you about a section like this, that would be the myocardium. And then if they ask you about a specific cell, that would be the cardiomyocytes. Okay, same thing here. A section, that would be the myocardium, and then the cells would be the developing cardiomyocytes. Another structure that might appear in the slides that you're given are these finger-like protrusions within the ventricle and within the aorta, and those are called the developing valves. If you remember from physiology, the valves act like doors that open and close depending on the pressure on either side of the chambers, either between the ventricle and the aorta or the atrium and the ventricle. And these valves, again, they are like small flaps or doors. Usually there are two of them, but again, because we're cutting it in a specific way, maybe one of them have been cut off and the other is more visible. So in this case, we can only see one here, and they are small flaps, or again, finger-like protrusions that goes into a developing valve or into a developing aorta. They might be these things right here. You can see them in other structures as well. For example, if this would be the developing aorta, this would be the developing valves. Before I continue, it's important to note that depending on the professor you're gonna ask, they're all gonna tell you a different thing, or they're all gonna disagree, I would say, on what sort of structure you're looking at when it comes to the atrium, the aorta, the truncus pulmonalis, and the vena cava superior. So these four structures are incredibly difficult to ascertain, I would say, or to identify, characterize under the microscope, because it's such a underdeveloped heart, and because these this image that you have is very 2D, and you'll not be able to see the full 3D context of the heart. So for example, the aorta here might appear here in another section of this slide, because it sort of slithers around the truncoconal region like so, right? Well, that is a horrible, it has this bow shape to it at least. That was my cat, excuse him. Some people might disagree about the naming of this being the aorta and think that this is more of an atrium. I've talked to my professor about it, and because it has this like bow-like shape, it's more in line of an aorta, and also the fact that we see developing valves. We don't really see these as clearly in the atrium. We see it more in ventricle. But some might also tell you that this is actually the aorta, professors and students alike. So ask your professor, talk to your professor, ask them about how to differentiate such structures. Because it's such a wishy-washy type of structure, unless it's very, very obvious in the slide that you'll get, I doubt they'll ask you about it during the colloquium. But I don't want to jinx it to you guys. It's at least not something that was super important for our colloquium. But again, not gonna jinx it. Another thing that is the most annoying part of the heart is the developing vena cava superior and the developing truncus pulmonalis. Now, these two blood vessels are, again, super hard to identify, and it's very difficult to be exact about them and know for sure that you're actually looking at them. This is my little tip on how to identify them. I'm gonna mention them. I don't think they're super important for the colloquium, but I'm gonna mention them. So if you've already identified the aorta, how do you identify the aorta? Well, it is usually the biggest blood vessel, and it has these valves in them, these finger-like protrusions, right? If you can identify the aorta, then you're looking at the rest of the truncoconal regions. And the blood vessels you see in the truncoconal regions, other than the aorta, if it's furthest to the left, so if we're looking at the image like this, right? If it's furthest to the left, it will be the vena cava superior. If it's furthest to the right, it would be the truncus pulmonalis. That is just what I imagined it, like how I imagined it. It might be completely wrong, so double check it with your professors, but this is just my tip, right? So we have the aorta here, we have a sort of finger-like protrusion there that looks like a valve, and it's directly connected to the ventricle. The furthest to the right would be the pulmonary artery. We can also see it here, right? So here we can correctly identify the developing aorta because we have these very identifiable valves. And because we only have one specific, or we have two blood vessels, the one furthest to the right, if this was positioned in like this, like it should look, or how it should look like, this would be the aorta, like I said, because we have the developing valves or finger-like protrusions there. So the one furthest to the right would be the truncus pulmonalis, and then the one furthest to the left, this could be a blood vessel, this could also be a blood vessel, would be the developing vena cava superior. Again, don't think this is super important for the colloquium. They might bring it in, but again, I think the most important part would be the developing aorta, maybe even the developing valves, but this whole entire region would just be called the truncoconal region, and it's more likely they'll ask you about the truncoconal region or ask you to write that rather than, you know, ask you about like a pseudo opening shape that could have been an artifact, which might be the vena cava superior, if you get what I mean. Okay. I think that's most of it. We have another set of structures. Of course, we have some surrounding blood vessels, like we have the developing veins here, but again, I don't think they're super important. Same with coronary artery developments. Again, take it with a grain of salt. Might not be important for the colloquium, but ask your professors. We also have these papillary muscle developments, which are at the bottom of the ventricles. We can see it there. Here we can't really see it. Here we can see it more clearly. They also look like valves. They're just towards the bottom of the ventricle. Can't really see it there. Oh, we can't. Here, these things. Okay, and these things. Perfect. Quick question. What, if you were to ascertain the age of this heart, what would you think this heart would be? 6 to 7 week old or 10 to 11 week old? If you said 6 to 7 week old, you would be correct. Why? Because we don't have a surrounding pericardium shawl like this, and also under the microscope, we can actually see the entire heart. So the heart hasn't been cut off here. We can actually see it in its entirety. Here also, the developing aorta might be this because of the developing valves. And then the furthest to the right, this would all be one thing, the truncus pulmonalis, and then the developing vena cava superior to the left. Hopefully that made sense. Coronary artery development, again, I don't think they're super important. If you see artery-like structures that are adjacent to the body of the heart, so not inside of the heart, but like next to it, they could be coronary arteries. Like these could be coronary arteries, maybe. The last structure I'd like to mention is the heart conductive system. Now I would say it's very hard to see it from here, so we have to see it more zoomed in an image like this one. Here we go. Here we go. So here we can see either the aorta or even maybe the ventricle or atria here, very big opening. And then we have some histology 2 structures here that might also be asked of you if the professor wants to test you for, with your skills from previous histology colloquiums. It's a slight chance, but it might happen. Then we have the, I would say this is more of the myocardium. There are some connective tissue in mesenchyme areas as well, but let's say this would be the myocardium with the cardiomyocyte cells. And then if you see a section that has been separated like this, so you can see that there is a border here that separates this section from the remaining section, right? And then the cells within it kind of looks like a big nerve fiber bundle. Obviously it's not the same, but it very much appears like a bundle of nerve fibers. These are developing Purkinje fibers, and the entire thing is called the heart conductive system. So in the colloquium, they might ask you about a specific section of the heart that aren't made up of the myocardium, that looks like it has been bordered like so, and has a different content of cells compared to the remaining area of cells. And that would be the heart conductive system. It's, I would say, more often than not, they'll ask you about the myocardium rather than the heart conductive system, just because it's very hard to actually find this. Same here. This is more of a zoomed in version of the heart, and in this case it's vena cava. Who is that? Vena cava superior. Let me get rid of that. And it's the wall of the vena cava superior before entering the heart, apparently. So here we have the wall, and of course the wall of any blood vessel can be split in like so, specifically muscle type blood vessels, I would say. Okay, that was the heart. Now we'll get into some other structures. I think within the abdomen, we might mention the lung for a second. Now, besides the heart, we also have one other major organ in the thoracical section, always followed up by these developing ribs, right? And that is the developing lung in pseudoglandular stage. You'll have to, unfortunately, write the entire name out because the developing lung appears, I think, in 4 different stages. Don't quote me on that. But the specific stage we're looking at, both in colloquium 1 and colloquium 2, will be the pseudoglandular stage. And therefore, this entire thing, if they ask you about it, will be this would be the name that you'll have to use. Now, the lung has all of these openings known as the developing bronchi. However, those are more colloquium 2 structures. This is just me trying to explain what these are, and they do appear much like these, or this structure, the metanephros, with all of these openings as well. The only real difference is the position. So getting down into the difference, here we go. So how to differentiate the lung from the metanephros. So the lung is, like I said, always in the thoracic region, so that it will be followed up by all of these developing ribs. You can see it here as well. While the metanephros is towards the abdominal region, or in the abdominal region. Another difference as well is the texture, or the 2 sections of the metanephros. So the metanephros can be divided into 2 sections. The first one being this darker section, or the darker outside, known as the cortex. And the second one being this lighter middle part, known as the medulla, or the developing medulla. You have to write, and the developing cortex here. Developing cortex. It's important to know these structures because they might be asked of you during the colloquium. The lung, on the other hand, doesn't have a lighter center and a darker cortex. It's the same color through and through and has the same texture through and through. While the metanephros, like we said, has a darker outside, known as the developing cortex, and a lighter medulla, known as the developing, or a lighter center, known as the developing medulla. Another structure that we can see on the thoracic region is the dorsal aorta. And the dorsal aorta can appear both in the thoracic area as well as the abdominal area. You can see it here. It's a large blood vessel, very, very large. It will appear large in the microscope as well, and can continue further down into the abdominal region, like you can see here. That was that. I think those are it for the thoracic area. We can finally get into the abdominal area and talk about these four structures more in depth. The developing liver is not a structure that is covered by colloquium 1, but these four definitely are. So let's start with the first one, or let's actually try to differentiate them first. So we have a developing adrenal gland. The developing adrenal gland is usually in comparison with these four. So we have the mesonephros, the developing gonad, the metanephros, which we just talked about, and the developing adrenal gland. Usually the developing adrenal gland is much larger than the remaining structures, and usually it's buddied up with the metanephros that we just talked about, that looks like the developing lung, with the darker cortex and the lighter middle. While the mesonephros is almost always accompanied by the developing gonad, and sometimes it can even be attached to the developing gonad. The mesonephros can appear sort of with these opening structures, or it can have like a completely homogeneous surface like the developing gonad. The only real difference, sometimes actually it's hard to differentiate the mesonephros from the gonad. The gonad can be smaller than the mesonephros in some slides. It can be larger, but this textures might be the same. The main difference that I'd say is that the gonad have these worm-like structures within it. So here we see the developing gonad. We also see the developing mesonephros. See how the texture of the mesonephros and the texture of the gonad is kind of the same, all except this specific structure that we can only see in the gonad. This is known as the primitive 6 cords, and they're made up of the primitive germ cells. While the gonad and the mesonephros are less important structures compared to the developing adrenal gland and the metanephros, they could still appear. So I'm just mentioning them just in case. But here you can see that the developing gonad is attached to the mesonephros right there. But the main difference is all of these primitive 6 cords that we can see within the developing gonad, almost appearing like these jelly worm structures, right? Jesus, all right. Someone got mad. While in the mesonephros, we don't really have those worm-like structures, but we might have these degenerating renal corpuscles in the mesonephros. We can see it here. So this would be the developing gonad. The developing gonad have these primitive 6 cords. In this case, it's harder to see, but we can kind of make them out right here. They kind of look like veins, right? While the mesonephros attached to the gonad sometimes have these degenerating renal corpuscles. So sometimes they can actually appear almost like a metanephros with all of these circles and all of these very much look like bubbles that are sort of boiling up inside of the mesonephros, known as the degenerating renal corpuscles. We can see them sometimes we can't, but here, for example, that would be the mesonephros fragment, and then this would be the degenerating renal corpuscles, and then the developing gonad, which is always next to it, with the primitive 6 cords that is used in order to distinguish the gonad from the mesonephros. Now to the actual 2 most important structures of the 4. The adrenal gland we can start with. Like I said, the adrenal gland is usually the biggest structure in the abdominal region, of the 4 that we are covering. Let's look at it without the annotations to make it a little bit easier. Well, let's actually first look at it from the sagittal cut. So here we can see that we have the abdominal region right here. We have the dorsal aorta there with some blood cells in the middle. we have the thoracic region with the developing lung in pseudoglandular stage because of all of these developing ribs, and the vertebral column that continues further down. If we look at the abdominal region here, we can see the metanephros, right? It's close to the, or close in texture to the lung, except it has the darker border known as the developing cortex and a lighter center known as the developing medulla. Next to the metanephros usually is the developing adrenal gland, and it's usually circular. Sometimes it can have like this mushroom cap or half moon shape. Sometimes it also appears as a pomegranate, so circular like a pomegranate and then a zigzag sort of tip, almost like a pomegranate or like a dumpling. We can see it further down here. Here we can see actually all 4 of the structures within the abdominal region of the embryo. So first of all, mushroom-like cap, that would be the adrenal gland or the developing adrenal gland. Usually the metanephros is a lot closer to the adrenal gland, but in this case it's here, and we can see the darker cortex and the lighter center being the developing medulla. We can also see the mesonephros, which has all of these like bubbling openings, looking like it's about to explode, right? It's about to wither away. That would be the mesonephros with the degenerating renal corpuscles. And then we have the protruding out of the mesonephros, the developing gonad, which have the primitive sex cords, but they're hard to see here. Okay, that was that. Here we can't see either. Here we can see all 4 of them also. So all 4 of them here together. We have the adrenal gland, in this case, a little bit more of a mushroom shape or mushroom cap shape. And then we have the very large very large metanephros with the darker cortex and the lighter medulla, different from the developing liver in pseudoglandular stage that has the same color throughout the structure. And then further down, we have the developing gonad. Why? Because we can see these primitive sex cords running through almost like blood vessels right here, like a network of blood vessels, right? And then attached to it will be the mesonephros. That is the final structure. And the mesonephros can, like we said, have different shapes and sizes and textures to it. But if it's accompanied by the gonad, the structure that is always attached to the gonad is usually the mesonephros. What I find is that they will probably ask you more about these 2 structures, the developing adrenal gland and the metanephros, instead of these 2. That is the developing gonad and the mesonephros connected to it. Okay, and now if we go down to look at the demo lab and lecture slides. So here we have a metanephros. Again, darker cortex, much lighter center or developing medulla. The outer part is called developing cortex. The inner part is called the developing medulla. These 2 are both developing adrenal glands because we have 2 of them, or during embryogenesis there are 2 of them. I think, yes. I think we do have 2 of them. Okay. Further down, we can see the metanephros as well, with the darker cortex and the lighter developing medulla. And here we can see actually the developing adrenal gland looking kind of like a balloon that is about to be drained off of its air, right? Here, this would be the knot, and this would be the little balloon string, right? Kind of sometimes has that appearance as well, but as long as you can kind of see that it's circular or mushroom cap shape, you'll know that it is the adrenal gland. Also, that it is right next to the metanephros. These are mesonephros, not metanephros. They very much look like livers, or not livers, lungs. I hope I didn't say liver instead of lung here. If I did, I apologize. But they do appear like lungs. They have this homogeneous texture all throughout, so they don't have a darker cortex or a lighter medulla. Therefore, this is more in line to be a mesonephros and not a metanephros. Like I said, the mesonephros can appear in various different ways. Here we have a more pomegranate shape of the adrenal gland. Like I said, the adrenal gland is usually the largest and most circular structure. So here we have that circular structure and then the zigzag tip of the adrenal gland, which is very characteristic of the adrenal gland. To the left, we can't really see the metanephros here. I believe it's maybe towards the right of this, but to the left of this, we actually have a gonad and a mesonephros. The gonad being this tongue-like shape that we see here with this little thing as well. And we can see the primitive sex cords all throughout this structure. So that's how we know that it's the developing gonad. While the mesonephros is the thing that is attached to the gonad with the degenerating renal corpuscles. We can see more of the adrenal gland here, circular in shape and a zigzag sort of top. Here is another one, circular in shape with a kind of zigzag top, sort of like a pomegranate. Here is the mesonephros with the degenerating renal corpuscles and the developing gonads. I think this would be maybe also developing gonads, but we can see the primitive sex cords more clearly there. These are a pair that we always see almost always together. That would be a pomegranate shape adrenal gland or developing adrenal gland, and this would be the metanephros with the darker cortex and the lighter developing medulla. We can also see it accompanied by this dorsal aorta because it's a large artery. It's a large blood vessel type of structure with some blood cells within, and we know that it is the dorsal aorta. More zoomed in version of the metanephros. Is this the metanephros or is it the lung? If you said the lung, you would be correct. We have the ribs here, so we are in the thoracic area, and then this area would be the lung in pseudoglandular stage, and then starting from here would be the abdominal area down here. We can actually see the developing adrenal gland right there because of the pomegranate shape. And that would be the continuation of it. I think we covered that. I think that's it in terms of those four within the abdominal area. There are some other structures within the abdominal area that is mentioned as well. For example, recanalization of hindgut, which is also appears as like a almost like a like dorsal aorta. It has similarities to it, and sometimes usually has two like lumens in it towards the end of the embryo. So this would be like where the anal canal would be, and then that would be the recanalization of hindgut. Not as important for us, but again, ask your professors to make sure. We can also see it here. So this would be where the anal canal would be, and then here would be the recanalization of hindgut. To sum the embryo and sagittal section up a little bit before we continue with the last structure that is separate from this. The embryo and sagittal section has two main divisions. It has the thoracic region and it has the abdominal region. The thoracic region has usually three organs that we're looking at: the lung in pseudoglandular stage, the dorsal aorta, and sometimes the developing heart. The ribs is what divides the thoracic area from the abdominal area, more specifically the diaphragm that goes all across the area like so. In the abdominal area, we have four different types of organs that are more in the spotlight: the adrenal gland, developing adrenal gland, the metanephros with the developing cortex and the developing medulla, the developing gonad, and the mesonephros, including the recanalization of hindgut. More importantly, we have the developing vertebral column with notochords, individual vertebra, the intervertebral or developing intervertebral discs, as well as the primary ossification centers, which we can't really see here. Another important thing are the developing spinal ganglion, which we can see sometimes. The final thing with the embryo and sagittal section that I want to cover, to repeat myself, are the immature skeletal muscle fiber bundles, which we will always see right next to the vertebral columns or right next to any sort of bone development. So we can see it kind of here, we can definitely see it here, and zoomed in, for example, if we zoom into this vertebral column and we look at this area, or if we zoom into this vertebral column looking at this area, we will have structures that look like this. So zooming into those areas, we will have these striations or layers, hair-like layers, that are all immature skeletal striated muscle fibers. And they can come in bundles like this. If they ask you about this, that would be a bundle. And then if they ask you about one specific single strand, that would be just a fiber. Finally, we get to our last slide of this colloquium, and it is the prostate. Thankfully, there aren't a lot of structures we have to know. The prostate is made up of these smooth muscle cells that in a microscope look very large and almost like hepatocyte, I would say. So they look like this. See how large and unique they look? Compare that with the smooth muscle cells of the human uterus further up. This was a mistake. Here we go. Compare that with the smooth muscle cells of the human uterus. This looks a lot more like muscle tissue than the prostate, right? The prostate cells that look a lot more like hepatocyte. These are this, even though they are the same cell. So these are smooth muscle cells. Another structure within the prostate are the prostatic glands. So they are scattered pretty evenly throughout the prostate. You can see them here as well. All of these are the prostatic glands. The largest prostatic gland is actually known as the prostatic urethra. So if they show you this under a microscope and they show you a pretty big prostatic gland-like shape and a smaller one, this would be the normal prostatic gland, and this would be the prostatic urethra, just the largest one you see in the microscope. If you have endothelial thin walls that contains blood cells, those would obviously be blood vessels. So they might ask you that in order to trick you or to see if you actually know the difference between a blood vessel and a gland. Like we said, the gland has simple cuboidal epithelium, while the blood vessels have endothelial cells. Those were all for the prostate. We can see them here as well. All of these being the prostatic glands, the smooth muscle cells making up the prostate, and the blood vessels with the thin endothelial cell wall. Here we can also see kind of capillaries, I would also say. And then you can see the glands more clearly here with the cuboidal or simple cuboidal epithelium right there. I do want to bring up the difference between a prostate, an umbilical cord, and a prostate and a human uterus. First of all, the prostate doesn't appear that often in the colloquium, not according to our professors, but still a lot of people make the mistake of mixing these two up when I'm tutoring. Okay, so for the prostate, they don't have this characteristic simple columnar amniotic epithelium like we do see with the umbilical cord right there. Another difference is that the prostate is made up of these large smooth muscle cells, while the umbilical cord is made up of these fibroblast-like cells, maybe even mesenchymal cells, not really, but all of this is the Wharton's jelly, and they very much do not look the same as these smooth muscle cells. Another difference is that the umbilical cord have very, very, very large muscle type blood vessels. You can see them here, 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 and here. While in the prostate, you don't really see that large blood vessels. You can see some smaller ones, but not this big, these big ones, right? And these big ones are what the umbilical cord mostly is composed of. The last difference is that the umbilical cord rarely, if ever, has like one gland max, right? So you have this remnant of allantois here, a gland-like structure because it has a simple cuboidal epithelium. Go back to class 2 if you've forgotten. And there's only one of them max, one of those glands max, right? Well, in the prostate, you have a lot of them scattered right around this specific structure. Okay, I think that's it for the prostate and umbilical cord. The difference between the prostate and the human uterus, like I said, the prostate is made up of smooth muscle cells, and the human uterus's myometrium is also made up of smooth muscle cells. The only difference is in the prostate, they're cut almost in a cross-section type, and they appear almost like hepatocytes, like large cells with very light cytoplasms and dark nuclei, while in the human uterus, these cells, although they might be zoomed in and appear more like these, zoomed out, they actually look very much like a characteristic view of muscle tissue. Another difference is that the glands of the human uterus is within the endometrium, not the myometrium. So while the glands of the prostate is within the entire slide, right? And they're surrounded by smooth muscle cells, while the myometrium of the human uterus don't have any glands. It's the endometrium, which doesn't have the smooth muscle cells, which have the glands. That's one difference. The most important difference is that the human uterus obviously has this border between the gland rich endometrium and the smooth muscle fiber tissue myometrium, which the prostate obviously does not. Okay, we talked about that. We could talk about the difference between umbilical cord versus fetal placenta, because this is the last class before the test yourself video. The umbilical cord have these large muscle type blood vessels, like we mentioned. The fetal placenta might also have it. You can see it here as well. You can see it here. However, the biggest difference is that the fetal placenta is always followed up by this area of tertiary villi. This here, and this here. While the umbilical cord does not have that area. Even though both are made up by simple columnar amniotic epithelium at the borders, the umbilical cord doesn't have those tertiary villi following up the slide. Another difference is obviously that the umbilical cord contains a gland within its structure, while the fetal placenta does not. Or sometimes it contains the remnant of a gland, right? We talked about the difference between a lung and metanephros, and that's it. Following up this video, we're going to do a test yourself video. I'm thinking 50-ish structures divided in 5 different attempts, 10 structures each. And then after each attempt, I'll go through why answers are correct, or the correct answers and why. Hopefully that'll help, and we'll see you in that video and in the colloquium 2 classes.